Hey guys, tonight we begin with something that might change everything you thought you knew about what makes us human. A tiny genetic change, just 804 letters of DNA, that may have given us our massive brains, but also made us sitting ducks for cancer. This isn't just another evolution story. This is the story of a biological trade-off that's still playing out in every one of our cells right now as you're watching this. So before you get comfortable, take a moment to like the video and subscribe, but only if you genuinely enjoy what I do here. And let me know in the comments where you're tuning in from and what time it is for you. It's always fascinating to see who's joining us from around the world. Now, dim the lights, maybe turn on a fan for that soft background hum, and let's ease into tonight's journey together. Picture this: you're sitting there, using that remarkable 3-pound organ inside your skull to process these words, to form thoughts, to imagine possibilities. Your brain has roughly 86 billion neurons, more than 3 times what our closest primate relatives possess. It's the organ that built civilizations, wrote symphonies, and sent rockets to the moon. But here's the thing that'll keep you awake tonight. The same genetic mutation that gave you this incredible thinking machine also made you vulnerable to a disease that kills nearly 10 million people every year: cancer. This isn't some cosmic coincidence. It's evolution's most dangerous bargain. And the story begins with a gene so small, so seemingly insignificant, that it went unnoticed for millions of years, until now. Deep in the laboratories where scientists peer into the very building blocks of life, researchers have uncovered something extraordinary: a gene called RGAP11B, just 804 base pairs long. To put that in perspective, your entire genome contains over 3 billion base pairs. This gene is a whisper in a symphony. And yet, this whisper may be responsible for everything that makes us uniquely human. But here's where it gets unsettling. That same genetic gift appears to be connected to our species' unusual vulnerability to cancer. While our closest relatives, chimpanzees, rarely develop the cancers that plague humans, we're sitting ducks. And scientists are beginning to understand why. The human brain is an evolutionary anomaly. It consumes 20% of our body's energy, despite being only 2% of our body weight. It requires an enormous skull that makes childbirth dangerous and infancy helplessly prolonged. From a survival standpoint, our brains should have been selected against. They're expensive, risky, and slow to develop. And yet, here we are. The secret lies in something called the neocortex. That's the outer layer of your brain, the part responsible for complex thinking, language, and consciousness. In most mammals, the neocortex is relatively small and smooth. In humans, it's massive and folded like a crumpled piece of paper, packed with neurons in every available space. For decades, scientists wondered what genetic switch had been flipped to create this neural explosion. The answer, it turns out, was hiding in plain sight. RGAP11B isn't ancient. It's not some primordial gene that's been with us since the dawn of life. This gene is young in evolutionary terms, appearing sometime after humans split from our common ancestor with chimpanzees about 6 million years ago. It's a duplicate, a genetic copy that went rogue and started doing something completely new. Here's what makes this gene so remarkable. When scientists introduced RGAP11B into mouse embryos, something extraordinary happened. Mice, which normally have small, smooth brains, began developing enlarged folded neocortices that looked strikingly human-like. The gene was literally building bigger brains. But the story gets darker when we dig deeper into how this gene actually works. RGAP11B doesn't just make more brain cells, it changes how cells behave at the most fundamental level. The gene sneaks into the mitochondria, the powerhouses of our cells, and alters their metabolism. It forces cells to consume glutamine, an amino acid, at unprecedented rates. This metabolic shift allows brain progenitor cells to divide rapidly and extensively, creating the vast neural networks that define human intelligence. But here's the problem. That same metabolic signature, that same glutamine addiction, that same rapid cell division, it's also the hallmark of cancer cells. Cancer, at its core, is a disease of cellular rebellion. Normal cells follow rules. They divide when they're supposed to, they die when they're supposed to, and they stay where they belong. Cancer cells break all these rules. They divide uncontrollably, refuse to die, and invade places they don't belong. And the metabolic changes that ARHGAP11B creates in developing brain cells are eerily similar to what cancer cells do throughout the body. Think about it. The same gene that gave us Shakespeare and Einstein may also be why humans have such high rates of cancer compared to other mammals. We traded longevity for intelligence. We traded cellular stability for cognitive complexity. This isn't the only genetic trade-off that made us vulnerable. Scientists have discovered another mutation that occurred during human evolution, this one affecting a protein called Fas Ligand. This protein is part of our immune system's arsenal against cancer. It's like a molecular assassin that hunts down and eliminates dangerous cells. But humans carry a unique mutation in this protein. Where other primates have a proline at position 153, humans have a serine. It sounds like a tiny change, but it's devastating. This single amino acid swap makes our cancer-fighting protein vulnerable to an enzyme called plasmin. Plasmin is found in high concentrations around aggressive tumors. It's like cancer's bodyguard, protecting tumors from immune attack. And because of our evolutionary mutation, plasmin can disable our Fas Ligand protein, essentially disarming our cellular police force. So not only did evolution give us genes that make our cells more likely to become cancerous, it also crippled one of our key defense mechanisms against cancer. This is evolution's cruel irony. The mutations that made us human also made us sick. But the story doesn't end there. Scientists are now discovering that this evolutionary vulnerability might actually hold the key to treating cancer. Understanding how ARHGAP11B works has opened new avenues for cancer research. If we can figure out how to disrupt the metabolic pathways this gene creates, we might be able to starve cancer cells of the glutamine they crave. And knowing about the Fas Ligand mutation has led to the development of new immunotherapies. Scientists are working on ways to protect this protein from plasmin, essentially restoring our immune system's ability to fight cancer. There's even more hope on the horizon. Researchers are studying animals that seem to have solved the cancer problem through different evolutionary paths. Elephants, for example, have multiple copies of a tumor suppressor gene called P53. They rarely get cancer despite their massive size and long lifespans. Naked mole rats have evolved a completely different strategy. Their cells produce a substance called high molecular weight hyaluronic acid that prevents tumors from forming in the first place. These discoveries are teaching us that cancer isn't inevitable. It's just the particular evolutionary path we took, and understanding that path might help us find our way out. The mitochondria in your cells right now are humming with the same metabolic processes that built your brain. The Fas Ligand proteins in your immune system are carrying the same vulnerability that has plagued our species for millennia. You are living proof of evolution's greatest experiment and its most dangerous gamble. But here's what gives me hope. We're the first species in Earth's history to understand our own evolutionary trade-offs. We can see the genetic bargains our ancestors made, and we're beginning to learn how to renegotiate the terms. The gene that gave us consciousness also gave us cancer. But consciousness, it turns out, might be exactly what we need to beat cancer. Every time you think a complex thought, every time you solve a problem or create something new, you're using the neural networks that ARHGAP11B helped build. The same gene that made us vulnerable also gave us the intelligence to understand that vulnerability. We're not just victims of our evolutionary history, we're its authors, and we're still writing the story. But this is just the beginning of understanding how our evolutionary past shapes our medical present. The relationship between brain development and cancer is just one piece of a much larger puzzle about what it means to be human in a biological sense. Speaking of puzzles that reshape how we think about human evolution, you need to see my video about the discovery that's rewriting the timeline of human civilization. Now let's dive deeper into the fascinating trade-offs that shaped human evolution. But first, take a moment to like this video and subscribe if you're enjoying this journey through our biological history. And let me know in the comments where you're tuning in from. It's always amazing to see who's joining us from around the world. The story we've uncovered so far reveals a profound truth about evolution. Every advantage comes with a cost. Our enlarged brains didn't just happen by accident. They were sculpted by millions of years of selective pressure, refined by countless generations who survived long enough to pass on their genes. But evolution isn't a perfect designer. It's more like a tinkerer, working with whatever materials are available, making compromises at every turn. Consider the metabolic burden of our massive brains. While our GAP11B helped expand our neural capacity, it also fundamentally altered how our cells process energy. This genetic innovation pushed our neural progenitor cells into a state of rapid division that mirrors what we see in cancer. It's a biological irony that the same cellular machinery that built our capacity for abstract thought, language, and civilization also left us vulnerable to one of our most feared diseases. The Fas Ligand mutation tells an equally complex story. This tiny change in our immune system may have been crucial for brain development, allowing neural cells to survive during the critical phases of growth when programmed cell death would normally eliminate them. But this same mutation made our cancer-fighting cells vulnerable to the very enzymes that tumors used to spread. It's as if evolution struck a bargain, expanded cognitive abilities in exchange for increased cancer susceptibility. We often think of natural selection as producing optimal solutions, but the reality is far messier. Evolution doesn't optimize for perfection. It optimizes for survival and reproduction in specific environments. Our ancestors faced immediate threats like predation, starvation, and infection. Cancer, which typically strikes later in life, would have been far less relevant to reproductive success than the cognitive advantages that helped our species dominate every ecosystem on Earth. This perspective helps explain one of the most puzzling aspects of human biology: why we're so much more cancer-prone than our closest relatives. Chimpanzees, despite sharing over 98% of our DNA, rarely develop the cancers that plague human populations. The difference lies in these evolutionary trade-offs. We gain the ability to create art, develop technology, and build complex societies. But we also inherited a genetic legacy that makes us vulnerable to diseases that were largely irrelevant to our ancestors' survival. The implications extend beyond individual health to how we think about medical research and treatment. Traditional cancer research has focused on finding and destroying malignant cells. But understanding the evolutionary origins of cancer susceptibility opens new avenues for intervention. If we know that certain cellular pathways were co-opted from brain development, we might find ways to selectively target cancer cells while protecting healthy tissue. The story of human evolution is ultimately a story about choices made by natural selection across vast timescales. Every adaptation that made us human came with hidden costs that we're only beginning to understand. Our big brains, our sophisticated immune systems, our capacity for language and culture. All of these remarkable features carry the signatures of ancient evolutionary compromises. But perhaps the most profound insight from this research is how it changes our relationship with our own biology. We're not perfectly designed machines. We're the products of a long, messy, imperfect process that made countless trade-offs along the way. Understanding these trade-offs doesn't diminish our achievements as a species. Instead, it helps us appreciate the remarkable journey that brought us to where we are today, and it gives us new tools for addressing the challenges we face. The mutations that gave us our remarkable brains also made us vulnerable to cancer. but they also gave us the cognitive capacity to understand these vulnerabilities and potentially to overcome them. In that sense, our evolutionary history isn't just a record of past trade-offs. It's a roadmap for future possibilities, written in the very DNA that makes us human. As we continue to unravel these ancient genetic bargains, we're not just learning about our past, we're gaining the knowledge we need to shape our future, one carefully targeted therapy at a time. But here's where this evolutionary tale takes a darker turn. While ARR-GAP11B was gifting us with bigger, more powerful brains, it was also setting us up for a collision course with cancer. Because the same metabolic pathways that made our neurons multiply so beautifully, they're eerily similar to the ones that cancer cells hijack when they go rogue. Remember that glutaminolysis we talked about? The way ARH-GAP11B forces cells to break down glutamine for energy? That's not just a brain development trick, that's cancer's favorite fuel source. When cancer cells want to grow fast, divide rapidly, and spread aggressively, they turn to the exact same metabolic processes that once built our magnificent minds. It's like evolution gave us a sports car with an engine that could outrun anything on the road, but forgot to mention that the same engine design makes the whole thing prone to catching fire. The mitochondrial changes that ARH-GAP11B triggers, the way it messes with cellular energy production, the rapid cell division it promotes, all of this reads like a blueprint for malignancy. And here's the truly unsettling part. The very regions of the brain that ARH-GAP11B helped expand, those areas packed with neurons and complex circuitry, they're also prime real estate for brain tumors. The neocortex, our crown jewel of cognitive evolution, happens to be where glioblastomas love to set up shop. It's as if the same genetic program that made us human also painted a target on the most important parts of our anatomy. But the story gets even more twisted when you look at the Fas mutation. Remember, this is the change that supposedly helped our brains get bigger by reducing premature cell death during development. Sounds great, right? Keep those precious brain cells alive, build a bigger, better thinking machine. Except there's a catch. Fas isn't just involved in brain development. It's one of our immune system's most important weapons against cancer. When your body detects a cell that's gone rogue, started dividing uncontrollably, or picked up dangerous mutations, Fas is part of the cleanup crew. It tells those cells to self-destruct, to die quietly before they can cause trouble. But the human version of Fas, thanks to that single amino acid change, has a fatal flaw. It's vulnerable to plasmin, an enzyme that tumors produce in abundance. So just when your immune system is trying to eliminate cancer cells, just when Fas is doing its job and telling the bad guys to die, the tumor fights back. It releases plasmin, chops up the Fasl protein, and essentially disarms your body's natural defense system. It's biological sabotage. Your immune cells show up to the fight, weapons drawn, ready to eliminate the threat. But the tumor has already figured out how to jam their guns. The very mutation that may have helped build our big brains also left us defensively naked against one of the most dangerous cellular enemies we face. And this isn't just theoretical. Clinical studies have shown that tumors with high plasmin activity are particularly aggressive, particularly resistant to treatment, particularly good at evading immune responses. The cancers that have learned to exploit this evolutionary weakness are often the ones that kill fastest. So here we are, walking around with brains built by genetic changes that also made us sitting ducks for cancer. Every time you have a complex thought, solve a difficult problem, or create something beautiful, you're using neural machinery that came with a hidden cost. Evolution didn't ask our permission for this trade-off. It just handed us incredible cognitive power and a cancer vulnerability, then walked away. This is the brutal mathematics of natural selection. There's no designer carefully weighing pros and cons, no safety inspector making sure the upgrades don't create new problems. There's just survival, reproduction, and the relentless pressure to pass genes to the next generation. If a mutation helps you live long enough to have children, evolution doesn't care what happens to you afterward. Our ancestors, who carried ARID-GAP11B and the modified Fas protein, probably did just fine. They had bigger brains, better problem-solving abilities, improved survival skills. They found food, avoided predators, attracted mates, raised families. The cancer risk? That mostly showed up later in life, after the reproductive years, after evolution had already moved on to the next generation. But now we live longer, much longer. We've extended human lifespan far beyond what our ancestors experienced, far beyond what evolution optimized for. And suddenly, those hidden costs are coming due. The same genetic changes that made us the dominant species on Earth are now stalking us in hospitals and oncology wards. This is why understanding our evolutionary history isn't just academic curiosity. It's a roadmap to our vulnerabilities, a guide to the biological booby traps we've inherited. However, ARID-GAP11B is not without consequences. Its metabolic demands mirror those of cancer cells. The gene drives glutaminolysis, the conversion of glutamine to glutamate, feeding the tricarboxylic acid cycle in ways that sound eerily familiar to oncologists. Cancer cells use this exact metabolic pathway to fuel their relentless growth. The same mitochondrial manipulation that allowed our brains to expand also created a cellular environment that cancer exploits. Think about this for a moment. The genetic innovation that gave us consciousness, creativity, and civilization also handed cancer a blueprint for survival. We engineered our own vulnerability into our DNA. The very metabolic processes that built the human mind became the foundation for malignant transformation. But the evolutionary trade-offs didn't stop there. Another mutation, this one in a gene called Fas Ligand, further compromised our cancer defenses. Fas Ligand is crucial for immune cells to eliminate threats, including cancer cells. It's the weapon our T cells use to destroy abnormal cells before they become tumors. But humans carry a unique mutation where serine replaces proline at position 153 in the protein. This seemingly minor change makes our Fas Ligand vulnerable to plasmin, an enzyme that aggressive tumors produce in abundance. When cancer cells release plasmin, it disables our immune system's primary weapon against them. It's like giving an enemy the key to your armory. The mutation that may have contributed to our larger brains simultaneously weakened our ability to fight the very cancers those brains would eventually develop. Chimpanzees don't have this vulnerability. Their Fas Ligand remains functional even in the presence of plasmin. This helps explain why our closest evolutionary relatives have significantly lower cancer rates than humans. We traded immune competence for cognitive complexity, and cancer found the gap in our defenses. The timing is crucial here. These genetic changes occurred after we split from chimpanzees, but before our brains reached their current size. They were essential steps in human evolution, but they came with hidden costs that only became apparent as we began living longer lives. For most of human history, these vulnerabilities didn't matter. People died young from infections, accidents, or violence long before cancer could develop. Modern medicine changed that equation. We conquered infectious diseases, improved nutrition, and extended lifespans. Suddenly, the evolutionary trade-offs that created our intelligence were exposed. The same cellular machinery that built our remarkable brains now had decades to malfunction, accumulate damage, and transform into cancer. This creates a profound paradox. The genetic innovations that made us human also made us uniquely susceptible to the diseases that now define aging in developed societies. Our intelligence allowed us to create the medical advances that extended our lives, but those extended lives revealed the hidden costs of our evolutionary success. Cancer in this light isn't just a disease, it's the shadow cast by our greatest evolutionary achievement. Every moment of human creativity, every scientific breakthrough, every work of art exists because of genetic changes that also fuel malignancy. We are simultaneously evolution's greatest success and its most vulnerable creation. Understanding these connections opens new therapeutic possibilities. If we know that HGAP11B drives both brain development and cancer-like metabolism, we can target those pathways more precisely. If we understand how plasmin disables our immune defenses, we can develop drugs to protect Fas Ligand or inhibit plasmin activity. Our evolutionary history becomes a roadmap for medical intervention. The story doesn't end with vulnerability, though. It ends with the remarkable fact that despite these genetic trade-offs, despite carrying cancer-promoting mutations in our DNA, most of us don't develop cancer. Our bodies have evolved additional layers of protection, backup systems, and repair mechanisms that usually keep the malignant potential of our own genetics in check. We are walking contradictions, biological marvels carrying both the seeds of genius and the potential for destruction in every cell. The same DNA that writes poetry and builds rockets also harbors the capacity for uncontrolled growth. Understanding this duality doesn't diminish human achievement. It makes it more precious, more miraculous, and more urgently important to protect. The hidden mutation that gave us big brains and fuels cancer reminds us that evolution doesn't optimize for perfection. It optimizes for survival long enough to reproduce. Everything else, including the diseases of aging, the cancers that emerge after decades of life, the slow accumulation of cellular damage, falls outside evolution's immediate concern. But we have something evolution doesn't: consciousness, the ability to understand our own biology, to recognize these trade-offs, and to intervene. The same brains that carry cancer-promoting mutations can now devise strategies to overcome them. We can outsmart our own evolutionary limitations, not by changing our DNA, but by understanding how to live with it, protect it, and when necessary, correct it. This is the true power of human intelligence. Not just the ability to create and discover, but the capacity to recognize our own vulnerabilities and transcend them. We may carry the seeds of cancer in our DNA, but we also carry the tools to fight back. Now that we've explored the fundamental mechanisms of our evolutionary trade-offs, we must confront a sobering reality about the modern implications of these ancient genetic changes. The same mutations that gifted us extraordinary cognitive abilities have created vulnerabilities that manifest most devastatingly in one of our greatest contemporary challenges: cancer in childhood. The cruel irony of our evolutionary legacy becomes starkest when we examine pediatric cancers. These diseases strike at the very population that should represent the pinnacle of our species' genetic optimization. Children, with their rapidly developing brains and growing bodies, should theoretically be the most protected by our evolutionary safeguards. Yet they are not. And this contradiction reveals the deepest consequences of the ancient bargain our species made. Most childhood cancers result from mutations in oncogenes and tumor suppressor genes acquired either during fetal development or shortly after birth. These aren't inherited defects passed down from parents, but rather spontaneous genetic errors that arise during the incredibly rapid cell division required for human development. The same cellular machinery that builds our enlarged brains, the same metabolic pathways that fuel our exceptional cognitive development, create windows of vulnerability that simply don't exist in our primate relatives. Consider the mathematics of human brain development. A developing human fetus must produce neurons at an average rate of 250,000 per minute during peak brain development. This is cellular division on a scale that dwarfs anything seen in chimpanzee development. Each division carries risk. Each replication of DNA creates opportunity for error. The ARHGAP11B gene that we discussed earlier doesn't just promote neural progenitor proliferation in healthy development. It creates an environment where cellular controls must walk an impossibly narrow line between growth and cancer. The metabolic demands of our developing brains compound this vulnerability. Human infants are born with brains that consume approximately 60% of their total energy expenditure, compared to just 8% in adult humans, and even less in other primates. This metabolic intensity requires cellular machinery to operate at the very limits of biological control. The same glutaminolysis pathways that HGAP11B exploits for brain expansion are the exact metabolic signatures that characterize many pediatric brain tumors. But the evolutionary perspective reveals why these vulnerabilities persist despite their obvious fitness costs. Until very recently in human history, childhood cancers were largely invisible to natural selection. Most children who developed these diseases would have died from other causes: infections, malnutrition, environmental hazards, long before cancer could manifest or progress. The diseases we now recognize as pediatric cancers existed, but they were masked by the brutal realities of pre-modern mortality. Natural selection, operating over hundreds of thousands of years, optimized our species for survival in environments where the immediate benefits of enhanced cognition, larger brains, more sophisticated social cooperation vastly outweighed the long-term risks of cancer. A child who survived to reproductive age with superior intelligence, enhanced learning capacity, and advanced social skills would have enormous advantages in passing on their genes. The possibility that they might develop cancer decades later, or that their children might face increased cancer risk, was irrelevant to evolutionary fitness in environments where most individuals didn't live past 30. This creates what evolutionary biologists call a life history trade-off. Our species invested heavily in early cognitive development and reproductive success, accepting increased long-term health risks as the price of short-term evolutionary advantage. The mutations that made us human were selected for their immediate benefits, not their long-term consequences. The Fas mutation we examined earlier provides another devastating example. This genetic change, which may have contributed to reduced cell death during brain development, left our immune systems vulnerable to cancer-promoting enzymes like plasmin. In children whose immune systems are still developing and whose bodies are undergoing rapid growth, this vulnerability is particularly pronounced. Pediatric cancers often exploit exactly these immune system weaknesses that our evolutionary history created. Modern medicine has revealed the full scope of this evolutionary legacy. We now live long enough, and our children survive long enough, to see the long-term consequences of genetic changes that were advantageous in our ancestral environment. We've extended human lifespan far beyond what our genes were optimized for, creating what researchers call an evolutionary mismatch between our biology and our environment. But here's the profound realization that transforms tragedy into hope. Understanding these evolutionary mechanisms gives us unprecedented power to intervene. We are no longer passive victims of ancient genetic trade-offs. We are the first generation of humans capable of recognizing, understanding, and potentially correcting the biological vulnerabilities that our evolution created. The same scientific understanding that revealed how HGAP11B promotes brain development is now informing research into how to protect developing neural tissue from cancer-promoting cellular environments. Researchers are exploring ways to maintain the cognitive benefits of rapid brain development while adding safeguards against the metabolic conditions that promote tumor formation. The discovery of the Fas mutation's role in cancer susceptibility has opened new avenues for immunotherapy research. Scientists are developing treatments that can protect the Fas protein from plasmin degradation, potentially restoring our immune system's ability to fight cancer, while preserving the neurological benefits that the original mutation provided. This represents something unprecedented in the history of life on Earth, a species becoming conscious of its own evolutionary constraints and developing the technological capability to transcend them. We are not just products of evolution anymore. We are evolution's partners, capable of continuing the optimization process that natural selection began, but with intention, compassion, and foresight that blind evolutionary forces could never possess. The children who face cancer today are not just victims of ancient evolutionary trade-offs. They are the motivation for humanity's next great evolutionary leap, the conscious design of our own biological future. Every research breakthrough, every new treatment, every life saved represents our species learning to master the very forces that created us. We began this journey in the darkness of our evolutionary past, where small genetic changes shaped the destiny of our species. The hidden mutation that gave us our extraordinary brains also handed us an unwelcome inheritance. A cellular vulnerability that makes us uniquely susceptible to one of our greatest fears: cancer. Tonight, we're diving deeper into this ancient trade-off, exploring not just how our brains grew so large, but why that same evolutionary gift turned our bodies into ticking time bombs. You see, the mutation we discussed, RGAP11B, didn't just expand our neocortex, it fundamentally rewired how our cells handle energy, growth, and death. And while this rewiring created the neural architecture for language, art, and abstract thought, it also created the perfect conditions for cellular rebellion. Let's talk about what happens inside a cell when AR8GAP11B goes to work. This gene doesn't just tell brain cells to multiply. It hijacks their entire metabolic machinery, forcing them into a state of rapid, almost frantic division. It targets the mitochondria, those tiny powerhouses inside every cell, and essentially puts them into overdrive. Under normal circumstances, mitochondria are remarkably well behaved. They produce energy, regulate cell death, and maintain a careful balance between growth and restraint. But AR8GAP11B changes the rules. It inhibits something called the mitochondrial permeability transition pore, a cellular checkpoint that normally prevents runaway growth. When this checkpoint is disabled, cells enter a metabolic state called glutaminolysis. They start consuming glutamine, an amino acid, at an extraordinary rate, converting it into fuel for rapid division. This is exactly what cancer cells do. They hijack the same metabolic pathways, exploit the same cellular machinery, and use the same molecular tricks that AR8GAP11B taught our brain cells millions of years ago. But the parallels go deeper than metabolism. Remember how AR8GAP11B expanded the population of basal radial glia, those stem cells that gave us our large brains? Cancer operates on the same principle. It takes normal cells and transforms them into something resembling stem cells, cells that can divide indefinitely, resist death signals, and colonize new territories. This isn't coincidence. It's the dark mirror of human evolution. The same genetic innovation that made us capable of contemplating the stars also made us vulnerable to the cellular chaos we call cancer. We didn't just inherit bigger brains from our ancestors. We inherited their molecular mistakes, and it gets worse. That second mutation we mentioned, the change in Fas Ligand, created an entirely different vulnerability. Fas Ligand is supposed to be our immune system's executioner, the protein that tells damaged or dangerous cells to die. It's particularly important for killing cancer cells before they can establish tumors. But our version of Fas Ligand has a fatal flaw. That single amino acid change, serine instead of proline at position 153, makes it vulnerable to an enzyme called plasmin. And here's the cruel irony. Many aggressive cancers produce plasmin in large quantities. They use it to break down the barriers between tissues, to invade new areas, to metastasize. So not only do these cancers spread more easily because of plasmin, They also disable our immune response by destroying the very protein meant to kill them. It's like giving a burglar the key to your house and then disabling your security system. This creates a perfect storm. Our ARH-GAP11B enhanced cells are already primed for rapid division and metabolic chaos. Our Fas Ligand is easily neutralized by the enzymes that aggressive cancers produce. And our extended lifespans, another gift of human evolution, gives cellular mutations decades to accumulate and compound. Other animals don't face this dilemma. Chimpanzees, our closest relatives, rarely develop cancer. Their cells divide more slowly, their Fas Ligand is resistant to plasmin, and they don't live long enough for most cancers to emerge. They traded away the possibility of Shakespeare and Einstein, but they also traded away the possibility of childhood leukemia and pancreatic cancer. But here's what's truly haunting about this evolutionary bargain. The children who suffer from cancer today are paying the price for innovations that occurred millions of years ago. Every pediatric oncology ward, every family devastated by a young life cut short, is connected to the same genetic changes that allowed humans to build civilizations, compose symphonies, and cure diseases. And yet, this story isn't just about ancient mistakes, it's about modern possibilities. Because understanding the evolutionary roots of cancer is opening up entirely new approaches to treatment. Scientists are developing therapies that target glutaminolysis, the same metabolic pathway that ARH-GAP11B exploited. Others are designing protective molecules that shield Fas Ligand from plasmin, restoring our immune system's ability to eliminate cancer cells. We're even learning from animals that solve these problems differently. Elephants, despite having thousands of times more cells than humans and living for decades, almost never get cancer. They've evolved multiple copies of p53, a tumor suppressor gene, and their cells are exquisitely sensitive to DNA damage. When an elephant cell detects even minor genetic problems, it immediately self-destructs. Some researchers are exploring whether we can give human cells similar capabilities. Others are investigating why naked mole rats, despite living in cancer-causing environments and having lifespans similar to mice, are essentially immune to cancer. Their cells produce a unique substance that prevents the cellular crowding that leads to tumor formation. These aren't just academic curiosities, they're blueprints for the future of cancer prevention and treatment. By understanding how evolution shaped our vulnerabilities, we're learning to engineer our way out of them. But perhaps the most profound lesson from this evolutionary story is about the nature of progress itself. Every advance comes with hidden costs. Every adaptation creates new vulnerabilities. The same neural complexity that allows us to love, create, and dream also makes us susceptible to depression, anxiety, and neurodegenerative diseases. The same immune system that protects us from infections can also attack our own tissues in autoimmune disorders. We are, in essence, evolutionary experiments. Magnificent, flawed, ongoing experiments in what it means to be conscious, creative, and mortal. Our susceptibility to cancer isn't a design flaw. It's the inevitable consequence of becoming human. And maybe that's okay, because the same species that inherited these ancient vulnerabilities is also the species that can understand them, study them, and ultimately transcend them. We may have been shaped by evolution, but we're no longer passive subjects of natural selection. We're active participants in our own biological destiny. The mutations that gave us big brains also gave us cancer. But those big brains are now working to solve the very problems they created. In laboratories around the world, researchers are unraveling the molecular mechanisms of cellular rebellion. They're developing immunotherapies that restore our natural defenses, targeted treatments that exploit cancer's metabolic weaknesses, and preventive strategies that could eliminate many cancers before they start. This isn't just about medicine. It's about completing the evolutionary journey that began millions of years ago, when a small genetic change started expanding human brains. We've used those brains to understand our own biology, to read the story written in our DNA, and to begin rewriting the parts we don't like. The ancient trade-off that gave us consciousness in exchange for cancer may not be permanent. We're learning to keep the gifts while discarding the curses, to maintain our cognitive abilities while eliminating our cellular vulnerabilities, to remain human while becoming something better than human. It's a profound thought. The same species that emerged from the African savanna with slightly larger brains and a tendency toward cancer may be the species that ultimately conquers death itself, not through evolution, but through intelligence, not by accident, but by design. The hidden mutation that made us human also made us mortal. But mortality, it turns out, may not be permanent. In the end, our big brains may be the key to unlocking immortality itself. The greatest irony of all would be if the genetic changes that made us vulnerable to cancer ultimately led us to defeat aging, disease, and death. That would be the final chapter in this evolutionary story. Not just survival of the fittest, but transformation of the fittest. Not just adaptation to environment, but mastery over biology itself. The mutation that gave us big brains may have also given us the tools to transcend every limitation that evolution ever imposed. And if that happens, if we truly learn to engineer our own biology, then every cancer patient, every family touched by this disease, every life cut short by our evolutionary inheritance, will have contributed to something magnificent. The final victory of consciousness over chaos, of intelligence over accident, of human will over the blind forces that created us. The hidden mutation that gave us big brains and fueled cancer may ultimately be remembered not as a curse, but as the first step towards something extraordinary. The beginning of our journey from evolved beings to self-designed ones. The hidden mutation that gave us big brains and fuels cancer lies buried deep in our DNA, a genetic gamble that shaped the very essence of what makes us human. But this isn't just another story about evolution and disease. This is about the moment our ancestors chose complexity over safety, intelligence over immortality, and how that choice still haunts our cells today. Tonight, we're diving into the shadowy intersection where genius meets genetics, where the same molecular switches that built our magnificent minds also opened the door to our most feared enemy. This is part 7 of our journey through the Slumber Lab, where the darkest secrets of human biology unfold under the cover of night. The mutation we're exploring tonight didn't just change our brains. It rewrote the fundamental rules of cellular life. And the price we pay for our cognitive supremacy is written in the language of cancer, whispered through our DNA, carried in every breath we take. But before we unravel this genetic mystery, let's understand what we're really talking about. When we speak of mutations that gave us big brains, we're not discussing a single dramatic change. We're talking about a cascade of genetic alterations that unfolded over millions of years, each one building upon the last, each one carrying its own risks and rewards. The most significant of these changes involves our cellular metabolism, the very engine that powers our thoughts. Our brains consume roughly 20% of our daily energy, despite making up only 2% of our body weight. This voracious appetite demanded evolutionary solutions that would prove to be a double-edged sword. At the molecular level, the mutations that enabled our large brains also altered how our cells handle energy, how they divide, and how they die. These same pathways, when disrupted, become the highways that cancer travels. The genetic switches that once allowed our neurons to proliferate and connect in unprecedented ways can be hijacked by malignant cells seeking their own twisted immortality. Consider the RGAP11B gene, a genetic sequence that exists only in humans and our closest extinct relatives. This tiny fragment of DNA, barely 800 letters long, emerged after we split from chimpanzees. When scientists introduced this gene into developing mouse brains, something extraordinary happened. The mice began growing larger, more folded brain tissue, complete with the neural structures that characterize human intelligence. But here's where the story takes a darker turn. The same cellular mechanisms that ARH-GAP11B triggers to expand brain tissue also mirror the processes that cancer cells exploit. The gene influences how cells divide, how they migrate, and how they consume energy. It pushes cellular metabolism toward a state that biologists call aerobic glycolysis, where cells burn sugar rapidly even in the presence of oxygen. This metabolic shift was crucial for brain development. Growing neurons need enormous amounts of energy, and this altered metabolic state provided the fuel necessary for our ancestors to build their increasingly complex neural networks. But cancer cells have discovered this same metabolic pathway. They hijack aerobic glycolysis to fuel their own rapid growth, creating the energy they need to divide relentlessly and spread throughout the body. The parallels don't end there. Brain development requires cells to ignore normal growth controls, to migrate to new locations, and to form connections in ways that would be considered pathological in any other tissue. Neural stem cells must proliferate rapidly, resist cell death signals, and sometimes even invade surrounding tissues to reach their final destinations. These are precisely the abilities that define cancer. It's as if evolution taught our brains the language of malignancy, and cancer learned to speak the same tongue. The cellular programs that sculpted our minds became the instruction manual for our most feared disease. But the relationship between brain evolution and cancer extends beyond metabolism. Another crucial mutation affects our immune system's ability to fight tumors. The Fas protein, which helps immune cells eliminate threats, carries a single amino acid change that makes it vulnerable to an enzyme called plasmin. This enzyme, elevated in many aggressive cancers, can disable our tumor-fighting cells just when we need them most. This genetic change may have provided advantages during brain development, possibly by reducing premature cell death in neural tissue. But in the context of cancer, it represents a critical weakness in our defenses. Tumors that produce high levels of plasmin can effectively disarm our immune system, explaining why certain cancers prove so difficult to treat. The mutation in Fas illustrates a fundamental principle of evolution. Changes that benefit one aspect of biology can create vulnerabilities elsewhere. Our ancestors didn't evolve to live long enough for cancer to become a major concern. Natural selection optimized our biology for survival and reproduction in a world where most people died young from infection, injury, or predation. In that environment, mutations that enhanced brain development and cognitive ability provided enormous survival advantages, even if they carried hidden costs that would only manifest decades later. The pressure to develop bigger, more capable brains was so intense that evolution accepted the cancer risk as an acceptable trade-off. This evolutionary bargain becomes even more complex when we consider the role of cellular senescence, the process by which cells stop dividing and eventually die. Our brains require some cells to resist senescence, to maintain their function across many decades of life. But this resistance to cellular aging creates opportunities for cancer to emerge. The mutations that extended the lifespan of our neurons also weakened the cellular safeguards that prevent malignant transformation. Brain cells that refuse to die when damaged, that continue dividing when they should stop, that resist the normal checks and balances of cellular life. These same properties define both neuronal resilience and cancer progression. Modern medicine has revealed just how deep these connections run. The same genes that regulate brain development, genes like p53, BRCA1, and mTOR, are also central players in cancer biology. Mutations in these genes can cause both neurodevelopmental disorders and increased cancer risk, highlighting the shared molecular machinery underlying brain function and malignant disease. Even our brain's unique energy demands created cancer vulnerabilities. The blood-brain barrier, evolved to protect our precious neural tissue from toxins and pathogens, also shields brain tumors from many cancer treatments. The very structure that safeguards our intelligence becomes a fortress that protects our most deadly brain cancers. The irony is profound. The mutations that lifted us above all other species, that gave us art and science and philosophy, that allowed us to contemplate our own existence and reach for the stars. These same genetic changes made us uniquely vulnerable to a disease that can steal everything we've become. But this story isn't just about the past. Understanding the connection between brain evolution and cancer opens new possibilities for treatment. If cancer exploits the same pathways that built our brains, perhaps we can use that knowledge to develop more effective therapies. Scientists are now exploring treatments that target the metabolic pathways shared by developing neurons and cancer cells. They're investigating ways to strengthen the immune defenses that our evolutionary history weakened. They're even studying how the brain's natural resistance to certain types of damage might be applied to cancer prevention. The hidden mutation that gave us big brains and fuels cancer represents one of evolution's most profound gambles. In choosing intelligence over immortality, complexity over safety, our ancestors set us on a path that would lead to both our greatest triumphs and our deepest fears. Tonight, as you drift off to sleep, consider the cellular legacy you carry. In every neuron that fires, in every thought that forms, in every dream that unfolds, you experience the wonder of a brain shaped by millions of years of evolutionary risk-taking. But in that same cellular machinery, in those same molecular pathways, lurks the shadow of cancer, waiting for the right combination of time and circumstance to emerge. The mutation that made us human also made us mortal, in ways our ancestors never imagined. But perhaps that's fitting. After all, it's our awareness of mortality, our knowledge of our own fragility, that drives us to seek meaning, to create beauty, to love deeply, and to reach beyond ourselves. In the end, the hidden mutation that gave us big brains and fuels cancer isn't just a biological curiosity. It's the genetic signature of what it means to be human, carrying both the promise of transcendence and the weight of consequence. And in that duality, in that eternal tension between light and shadow, we find not just the story of our evolution, but the essence of our humanity itself. In the ancient battlefields where arrows whistled through the air and swords clashed against shields, there existed a weapon that struck not with metal, but with molecules. An invisible killer that could topple kingdoms without leaving a single wound. Poison. The silent emperor of warfare that could eliminate enemies before they even knew they were under attack. Yet today, as we delve deeper into the story of human evolution, we encounter a far more complex poison. One that flows not through goblets or blade tips, but through our very DNA. A genetic mutation so small, so seemingly insignificant, that it can be measured in single letters of our genetic code. But this microscopic change carries with it the power to build empires of neurons and simultaneously construct the scaffolding for our own destruction. The irony is breathtaking. The same genetic innovation that lifted us from the African savanna and placed us among the stars also planted the seeds of cancer deep within our cellular machinery. It's a Faustian bargain written in the language of adenine, thymine, guanine, and cytosine. We traded immortality for intelligence. We exchanged cellular perfection for consciousness itself. But this isn't a story of defeat. It's a story of triumph shadowed by consequence. Because understanding this ancient trade-off, this primordial poison that courses through our veins, offers us something our ancestors never had: the possibility of rewriting the contract entirely. The mutation we're exploring tonight didn't just change how our brains developed. It fundamentally altered the metabolic machinery of our neural cells, rewiring them to consume energy at rates that would make a Formula One engine seem efficient. This hyperdrive cellular metabolism, this biological overclocking, enabled the rapid construction of billions of additional neurons. It allowed our ancestors to build brains capable of language, art, science, and love. But evolution never gives without taking. And what it took was our cellular discipline. The same metabolic pathways that fuel our thoughts, our dreams, our capacity to contemplate the universe itself, also provide the perfect environment for rogue cells to multiply without restraint. Cancer cells, it turns out, are simply normal cells that have rediscovered the metabolic strategies our brain cells use every day. The research revealing this connection reads like a biological detective story. Scientists studying the Rh-GAP11B gene, a tiny snippet of DNA that exists only in humans and our closest extinct relatives, discovered that this genetic newcomer doesn't just build bigger brains. It transforms the entire energy economy of developing neural tissue. Cells expressing this gene begin consuming glutamine at ravenous rates, feeding it into their cellular powerhouses to fuel unprecedented growth and division. Sound familiar? It should, because this is precisely the metabolic signature that oncologists have learned to fear. The Warburg effect, named after the scientist who first described cancer's unusual appetite for specific fuels, isn't actually a cancer phenomenon at all. It's a brain development phenomenon that cancer has learned to exploit. The implications are staggering. Every time you form a memory, every moment of creativity, every flash of insight, you're utilizing cellular machinery that operates on the same principles that drive malignant transformation. Your thoughts are powered by the same metabolic engines that tumors hijack for their relentless expansion. But the story grows darker still. This genetic gift that expanded our minds also sabotaged our immune defenses in ways we're only now beginning to understand. Another mutation, this one affecting a protein called Fas Ligand, has left humans uniquely vulnerable to certain types of cancer compared to our closest living relatives. Chimpanzees, our evolutionary cousins who diverged from our lineage just 6 million years ago, possess immune systems that can efficiently eliminate many of the cancers that plague human populations. Their Fas Ligand protein remains intact, capable of instructing damaged or dangerous cells to self-destruct. But in humans, a single amino acid substitution has rendered this crucial protein vulnerable to an enzyme called plasmin. Plasmin, normally involved in wound healing and blood clot dissolution, becomes a weapon in the hands of aggressive tumors. These cancers have learned to produce plasmin in abundance, using it to disable the very immune system components designed to destroy them. It's as if we evolved sophisticated locks for our cellular security system, only to hand the keys to our enemies. The research documenting this vulnerability reads like a catalogue of evolutionary trade-offs. The same genetic changes that allowed our brains to reach unprecedented size and complexity also created gaps in our cellular defenses. We became smarter, but we also became more fragile. We gained consciousness, but we lost a measure of our biological resilience. This isn't speculation or theoretical biology. The evidence is written in our genomes, documented in our disease rates, and visible in the laboratory studies comparing human and chimpanzee immune responses. When researchers expose human and chimpanzee immune cells to the same cancer-promoting conditions, the differences are stark. Chimpanzee cells maintain their defensive capabilities. Human cells, compromised by plasmin-sensitive Fas Ligand, fail to mount effective responses. The evolutionary timeline makes the tragedy even more poignant. These mutations didn't occur in isolation. They emerged during the critical period when our ancestors were developing larger brains, more complex social structures, and the cognitive abilities that would eventually lead to agriculture, civilization, and technology. The genetic changes that made us human also made us vulnerable. But perhaps most remarkably, these vulnerabilities were acceptable trade-offs for most of human history. Cancer, while present in ancient populations, rarely had the opportunity to manifest fully. Our ancestors faced far more immediate threats: predators, starvation, infectious diseases, and the harsh realities of survival in unforgiving environments. Most humans died long before the age when cancer typically strikes. The mutations that predisposed us to malignancy were essentially invisible to natural selection. They provided massive advantages during our reproductive years: enhanced brain development, improved cognitive abilities, better social coordination, while imposing costs that typically emerged only after we had passed our genes to the next generation. It's a cruel irony that our evolutionary success has created the conditions for these ancient vulnerabilities to manifest. Modern medicine has eliminated many of the threats that historically claimed human lives before cancer could develop. We've extended human lifespan far beyond what our ancestors experienced, inadvertently creating space for these primordial genetic time bombs to detonate. The research into metabolic reprogramming in both brain development and cancer has revealed just how deeply intertwined these processes truly are. The same pathways that drive neural proliferation during fetal development, the cellular mechanisms that construct the neural networks underlying consciousness itself, can be reactivated in adult tissues to drive malignant transformation. Scientists studying this connection have identified specific molecular switches that control the transition between normal cellular metabolism and the hyperactive energy consumption characteristic of both developing brain tissue and cancer cells. These switches, evolutionarily ancient and remarkably conserved, suggest that the capacity for rapid cellular growth and division has been a fundamental feature of life for hundreds of millions of years. What made humans unique wasn't the invention of these metabolic pathways. It was our willingness to use them extensively in brain tissue to push neural development to extremes that no other species had attempted. We became metabolic daredevils, cranking our neural machinery to levels that bordered on the dangerous, all in service of building brains capable of transforming the world. The telomerase connection adds another layer to this story. Telomerase, the enzyme responsible for maintaining chromosome ends during cell division, plays crucial roles in both brain development and cancer progression. During the critical period of human brain expansion, telomerase activity in neural stem cells must have been extraordinary, enabling the massive number of cell divisions required to build our oversized brains. But telomerase activation, while essential for brain development, also provides cancer cells with one of their most dangerous capabilities: the ability to divide indefinitely. Normal adult cells have limited replicative potential, a built-in safety mechanism that prevents runaway cellular proliferation. Cancer cells bypass this restriction by reactivating telomerase, essentially stealing the same immortalization machinery that builds our brains. The research documenting this connection spans decades and involves thousands of scientists across multiple continents. Yet the story it tells is remarkably consistent. Human evolution involved a series of calculated risks, genetic gambles that traded long-term cellular stability for short-term cognitive gains. These weren't random mutations or evolutionary accidents. They were systematic modifications to fundamental cellular processes, changes that required precise coordination across multiple genetic systems. The probability of such coordinated evolution occurring by chance approaches zero. Instead, what we see is evidence of intense evolutionary pressure, circumstances that made enhanced cognitive abilities so valuable that they justified accepting increased cancer risk. What were those circumstances? The fossil record and archaeological evidence point to a period of extreme environmental instability in Africa around the time these mutations emerged. Climate change, volcanic activity, and shifting ecosystems created conditions where cognitive flexibility became a matter of survival. Our ancestors faced challenges that couldn't be solved through physical strength or traditional adaptive strategies. They needed to think their way out of extinction. The genetic changes that enabled this cognitive revolution weren't implemented gradually. Molecular clock analyses suggest they occurred in a relatively compressed timeframe, evolutionarily speaking. This rapid implementation required accepting certain trade-offs, including the metabolic and immunological vulnerabilities that predispose us to cancer. But understanding these trade-offs also reveals their potential solutions. If cancer represents the dark side of the same cellular machinery that builds our brains, then studying brain development might provide insights into controlling malignant transformation. Researchers are already exploring this connection, investigating whether compounds that regulate neural metabolism might also influence cancer progression. The implications extend beyond individual treatments to species-level considerations. As we develop technologies that could potentially modify human genetics, understanding the full consequences of our evolutionary trade-offs becomes crucial. Do we accept the increased cancer risk as the price of consciousness? Or do we attempt to engineer solutions that preserve cognitive capabilities while restoring cellular stability? These questions aren't merely academic. Gene therapy technologies are advancing rapidly, and the possibility of modifying the very mutations that make us human is moving from science fiction to clinical reality. But such interventions require understanding not just the mechanisms involved, but the evolutionary logic that created them. The story of ARSH-GAP11B and Fas Ligand isn't just about two genetic mutations. It's about the fundamental nature of evolutionary trade-offs and the price of consciousness itself. Every human alive today carries the genetic legacy of ancestors who chose intelligence over immortality, consciousness over cellular perfection. We are the descendants of beings who looked at the universe and decided that understanding it was worth dying for. They traded genetic stability for the ability to contemplate the stars, to create art, to love, to suffer, and to search for meaning in the vast cosmic darkness. They chose consciousness, knowing it came with a price. That price is written in our DNA, encoded in every cell, manifest in every cancer diagnosis. But it's also visible in every scientific discovery, every work of art, every moment of human connection and understanding. The same genetic changes that make us vulnerable to malignancy also make us capable of studying that vulnerability, of understanding its origins, and potentially of transcending it. The research continues, driven by scientists whose own enlarged brains, products of these ancient genetic modifications, work to understand and ultimately overcome the limitations they inherited. It's a profound irony that the same cognitive abilities that cancer made possible are now being turned against cancer itself. Modern oncology represents humanity's attempt to renegotiate the evolutionary contract our ancestors signed. Through science, technology, and medical intervention, we're working to retain the cognitive benefits of our genetic modifications while minimizing their dangerous side effects. We're trying to have our evolutionary cake and eat it too. The success of this endeavor remains uncertain. Cancer continues to claim millions of lives annually, and the fundamental metabolic similarities between brain development and malignant transformation suggest that completely eliminating cancer risk might require sacrificing some of the cognitive capabilities that define us as human. But perhaps that's not the right goal. Perhaps the lesson of our evolutionary history isn't that we should eliminate all trade-offs, but that we should make them consciously and with full understanding of their consequences. Our ancestors made their choice in the darkness of genetic uncertainty. we have the privilege of making ours in the light of scientific understanding. The mutations that built our brains and fueled our vulnerability to cancer represent one of the most audacious gambles in evolutionary history. They transformed a lineage of African apes into a species capable of leaving the planet, of manipulating matter at the atomic level, of composing symphonies and solving mathematical theorems. They also condemned us to carry within our cells the seeds of our own destruction, molecular time bombs that can detonate decades after birth, turning our own tissues against us. It's a price our species has paid for millions of years, often without even understanding what we were paying for. But now we know. Now we understand the bargain our ancestors struck and the genetic legacy they left us. And with that understanding comes the possibility of choice. We can accept the trade-offs they made, grateful for the consciousness they purchased with their cellular stability. Or we can work to transcend those limitations, to engineer solutions that preserve the benefits while minimizing the costs. Either path requires acknowledging the profound courage of our evolutionary predecessors. They faced an uncertain future, armed only with the knowledge that enhanced cognition improves survival. They couldn't have known that their genetic modifications would eventually enable their descendants to travel to the moon, to decode the structure of DNA, or to contemplate the origin of the universe itself. They simply chose intelligence, wherever it might lead. And it led to us, a species capable of understanding its own evolutionary history, of recognizing the trade-offs that created it, and potentially of rewriting the genetic contracts that govern its future. The story of human evolution isn't finished. The same cognitive abilities that emerged from our ancestors' genetic gambles continue to drive scientific progress, medical advances, and technological innovations. We stand at a unique moment in history, possessing both the knowledge to understand our evolutionary vulnerabilities and the tools to potentially address them. The mutations that built our brains may have also built our cancers, but they also built our capacity to fight back. Every oncologist, every researcher, every person touched by cancer who chooses to support research rather than surrender to despair is participating in humanity's ongoing evolution. In our species' long journey from genetic vulnerability toward conscious control over our biological destiny. We are no longer passive recipients of evolutionary trade-offs. We have become active participants in our own genetic future. And that transformation, that shift from evolutionary subjects to evolutionary agents, may be the most remarkable consequence of the ancient mutations that made us human. The price of consciousness was cancer, but consciousness also gave us the power to understand that price and potentially to change it. But these weren't just theoretical vulnerabilities waiting to be discovered. They were active, dangerous trade-offs that shaped the very essence of what made us human. The same genetic modifications that allowed our ancestors to develop complex reasoning, language, and abstract thought also created cellular environments that cancer could exploit with devastating efficiency. Consider the metabolic demands of our enlarged brains. Human brains consume roughly 20% of our total energy, far more than any other primate. To fuel this cognitive powerhouse, our cellular machinery had to become incredibly efficient at processing glucose and managing energy production. But here's the dark irony. These same metabolic pathways that keep our neurons firing are precisely what cancer cells hijack to fuel their own rapid growth and spread. The ARH-GAP11B gene didn't just expand our neocortex in isolation. It fundamentally altered how our neural progenitor cells handled energy, how they responded to growth signals, and how they managed cellular division. These changes created a template that would echo throughout our bodies, establishing patterns of cellular behavior that, while beneficial for brain development, would later become cancer's blueprint for survival. But the story gets even more complex when we examine the immune system vulnerabilities, that came packaged with our cognitive gifts. The Fas Ligand mutation we discussed earlier wasn't just a random genetic accident. It appears to have been intimately connected to the same evolutionary pressures that drove brain expansion. The protein changes that made our immune cells less effective against certain cancers may have been necessary to prevent our developing brains from being attacked by our own immune systems during critical growth periods. Think about it this way. Our ancestors faced a brutal evolutionary calculus. Those genetic changes that enabled complex thought, planning, and social cooperation provided such massive survival advantages that they were worth the increased cancer risk. In a world where most humans died from predation, starvation, or infectious disease before age 40, the long-term consequences of cancer susceptibility were largely invisible to natural selection. The cellular mechanics tell a fascinating story. Modern research has revealed that many of the genes involved in human brain development also play crucial roles in cancer progression. The same molecular switches that allow neural stem cells to proliferate rapidly during fetal development can be reactivated by tumors centuries later. It's as if evolution wrote the same instruction manual for building bigger brains and for cancer cells to exploit our bodies. Even more unsettling is the realization that some of our most distinctly human traits may have come with the steepest cancer costs. Our extended lifespans, delayed reproduction, and complex social structures all require sophisticated cellular repair mechanisms and tightly controlled growth patterns. But these same systems, when they malfunction, create the perfect conditions for malignant transformation. The research emerging from comparative oncology paints a stark picture. Humans develop certain types of cancer at rates far exceeding our closest primate relatives. Brain tumors, in particular, show patterns that seem directly linked to the genetic changes that enabled our cognitive revolution. We didn't just evolve bigger brains, we evolved brains that were more vulnerable to the very cellular chaos that defines cancer. What makes this evolutionary trade-off particularly poignant is its timing. For most of human history, these genetic vulnerabilities remained largely hidden. Our ancestors lived in small groups, faced constant environmental threats, and rarely survived long enough for cancers to develop and spread. The same cognitive abilities that made us vulnerable to cancer also helped us develop agriculture, medicine, and social structures that dramatically extended our lifespans. In essence, we built a world where we could live long enough to experience the full consequences of our evolutionary bargain. Our success as a species created the conditions where our genetic vulnerabilities could finally express themselves. We became victims of our own cognitive triumph, living long enough in safe enough environments for the cancer time bombs embedded in our DNA to finally detonate. This isn't just academic speculation. Modern cancer research has identified dozens of genes involved in human brain evolution that also play roles in tumor development. The pathways that allowed our ancestors to develop language, abstract reasoning, and complex social behaviors are the same pathways that modern oncologists target when treating aggressive cancers. But perhaps the most remarkable aspect of this story is what it reveals about human nature itself. We are a species that quite literally thought our way into existence, developing cognitive abilities so powerful that they transformed not just our behavior, but our biology. The price we paid was written into our cellular code, a genetic mortgage that we're still paying today. Understanding this evolutionary context doesn't just change how we think about cancer. It transforms our entire understanding of human vulnerability and resilience. We're not just fighting a disease, we're grappling with the biological consequences of becoming human. Every cancer diagnosis, every tumor, every malignant cell carries within it an echo of the same genetic changes that allowed our species to contemplate the stars, create art, and unlock the secrets of our own biology. The scientists working to understand these connections aren't just studying cancer. They're decoding the biological history of human consciousness itself. As the metabolic machinery that had once powered our unique cognitive revolution began to demand its price, the very pathways that enabled human consciousness to flourish also carved channels for our greatest biological vulnerabilities. The glutaminolysis that feeds neural proliferation, the mitochondrial adaptations that fuel complex thought, the immune modifications that protected developing brains. All of these evolutionary gifts carried hidden costs that would only become apparent as human lifespans extended far beyond what our ancestors experienced. But perhaps the most profound irony lies not in what these mutations took from us, but in what they revealed about the nature of evolutionary trade-offs themselves. Every biological system exists in delicate balance, and the same genetic changes that lifted humanity above other species also tethered us to new forms of suffering. The ARHGAP11B gene that expanded our neocortex didn't just give us art, language, and philosophy. It gave us a metabolic signature that cancer cells would later exploit. The Fas cell mutation that may have protected our developing brains from premature cell death also handed tumors a weapon against our immune defenses. These weren't design flaws. They were the inevitable consequences of evolution's relentless optimization for survival and reproduction in environments our ancestors faced, not the ones we inhabit today. Natural selection shaped our biology for lives that rarely extended beyond 40 years, where cancer was far less likely to manifest, where the benefits of enhanced cognition and brain development far outweighed the long-term risks of metabolic vulnerabilities. The story of human evolution and cancer susceptibility forces us to confront uncomfortable truths about our place in the biological world. We are not perfectly designed organisms, but rather the products of countless genetic compromises, each one reflecting the specific pressures our ancestors faced. The very mutations that made us human, that gave us the capacity for abstract thought, cultural transmission, and technological innovation, also made us uniquely vulnerable to diseases that barely troubled our evolutionary cousins. Yet within this sobering reality lies extraordinary opportunity. Understanding these ancient trade-offs opens new avenues for therapeutic intervention. If we know that human cancer cells exploit the same glutaminolysis pathways that once fueled brain expansion, we can design treatments that target these metabolic vulnerabilities. If we understand how the Fas mutation makes our immune system susceptible to plasmin-mediated inactivation, we can develop strategies to protect or enhance immune function in cancer patients. The research emerging from laboratories around the world suggests that our evolutionary history, rather than being merely an academic curiosity, may hold keys to some of our most pressing medical challenges. Scientists are already exploring whether plasmin inhibitors could enhance immunotherapy effectiveness in solid tumors. Others are investigating whether targeting the metabolic pathways that both fuel cognition and feed cancer could lead to more precise, less toxic treatments. Perhaps most remarkably, the same technologies that allowed us to uncover these ancient genetic trade-offs, the sequencing technologies, computational biology, and molecular techniques that revealed ARHGAP11B's role in brain evolution and Fas' vulnerability to plasmin, now offer us tools to address the consequences of these evolutionary choices. Gene therapy, immunotherapy, and precision medicine all represent attempts to rewrite the biological scripts that evolution handed us. The hidden mutation that gave us big brains and fuels cancer is ultimately a story about the complexity of biological systems and the unintended consequences of evolutionary success. It reminds us that every advance comes with costs, every adaptation carries trade-offs, and every solution creates new problems to solve. But it also demonstrates the remarkable power of human curiosity and ingenuity to understand these systems, and perhaps to transcend some of their limitations. As we stand at the threshold of an era where genetic engineering and advanced therapeutics might allow us to address some of these ancient vulnerabilities, we carry with us both the burden and the gift of our evolutionary heritage. The same cognitive abilities that make us susceptible to certain cancers also give us the capacity to understand why, and potentially to do something about it. The mutation that made us human continues to shape our destiny, but for the first time in our species' history, we are no longer passive recipients of its effects. We are becoming active participants in our own evolution, using the very intelligence it gave us to address the vulnerabilities it created. In the end, perhaps that is the most human response of all, to take the hand we were dealt by evolution and play it with everything we've learned along the way.